Greetings everyone! This short video will cover the biological topics of evolution and speciation. In particular, we will define evolution, construct and examine cladograms as a method of illustrating evolution, and we will compare and contrast different species concepts. To begin, let's think about what evolution actually means. Simply put, evolution is change in the gene pool over time. It's important to remember that evolution does not happen to individual organisms, but rather happens to populations. As time progresses, this population of mice is likely to change. Over several generations, the mice on average might get bigger. They might get smaller. Or the mice might get faster. Whatever traits are more advantageous for survival and reproduction should appear more often in the population, which changes the gene pool of that population over time. You can probably imagine that, if given enough time, one population of mice might amass enough changes in its gene pool to make it very different from other populations of the same mouse species. When this happens, it is known as speciation, and the process is often depicted on diagrams like these, known as cladograms. Cladograms are branching diagrams that illustrate the evolution of organisms over time. When enough changes amass so that organisms diverge or speciate, the cladogram splits. So the branching pattern of the cladogram illustrates the evolutionary history of the organisms as they change over time. In this hypothetical cladogram, the evolutionary history of organisms A through E is depicted. To fully understand how cladograms work, it is important to see the basic structure of a cladogram as being composed of nodes and branches. Branches are the lines that indicate a lineage of organisms through time. Nodes are the point at which a branch splits and illustrate two species or groups diverging from one another. Now that we've covered the structure of a cladogram, let's discuss the proper way to read a cladogram. You should start by following the cladogram from the tips of the branches down the trunk and then back again to understand the pattern of the nodes and the branches. Without fully examining the branching pattern, it might be easy to mistakenly interpret the relationships of the organisms. For example, organisms C and D are adjacent to one another on the cladogram. So you might think that organism C is more closely related to organism D than it is to organism E. But that isn't the case. Both organisms D and E are removed from C by essentially the same nodes and branches. So it won't make a difference if the cladogram is constructed like this. Likewise, rearranging the cladogram like this also does not change the essential branching pattern and therefore does not change the depiction of the evolutionary history of these organisms. Cladograms can be constructed in a variety of shapes and formats, but they can all be interpreted in the same way. Organisms or groups that share a node closer to the tips of the tree are more closely related than those sharing a node further down the trunk. So, if we return to the mice, one question you might have is, how do we know when speciation has occurred? How many changes are necessary to create this split in the cladogram? The answer, which is the bane of biology students everywhere, is, it depends. More specifically, it depends on which species concept you are applying. The biological species concept is one of the most frequently applied species concepts. It states that if organisms are able to reproduce and create viable fertile offspring, then they are the same species regardless of any differences in morphology, physiology, or geography. If, however, one population of these mice has changed to the point where it is no longer willing and or able to reproduce with other populations, then we can consider it a different species that has diverged from the original lineage. The primary problem with the biological species concept, though, is that it can't be applied to organisms that reproduce primarily via asexual means, like bacteria. Because most bacteria rely on binary fission for reproduction, the biological species concept doesn't exactly apply. So then, how do we know when speciation in bacteria or other asexually reproducing organisms has occurred? To answer that question, let's consider another asexually reproducing organism that is getting plenty of attention in the media today: vampires. Technically speaking, vampires reproduce asexually because they make more vampires simply by biting people and turning them into new vampires. So, how can we tell if speciation among vampires has occurred? We must look for unique heritable characteristics that are present in a population of vampires but not in others. For example, imagine that there is a genetic mutation that allows a vampire to survive in sunlight. That characteristic is advantageous and should allow that vampire to feed and reproduce more easily. And he should pass that trait along to his offspring, creating a population of vampires that is ready for a day at the beach. This is what is known as the phylogenetic species concept. When a population has unique heritable traits that are not found in other populations, then we can consider them to be a different species. This concept is often applied to asexually reproducing organisms, like vampires and bacteria, but it can also be applied to sexually reproducing organisms as well. In reality, biologists often use both the biological species concept and the phylogenetic species concept to construct cladograms. These two different concepts are based on different data, reproductive isolation versus uniquely heritable traits, so biologists will often take advantage of both to ensure that the cladogram illustrates the evolutionary history of the organisms as accurately as possible. To conclude, let's summarize what we've covered in this video. Evolution is a change in the gene pool of a population over time, and this evolutionary change can be depicted by the branching pattern of cladograms. Finally, different species concepts can be useful for different types of organisms. The biological species concept is often applied to sexually reproducing organisms, while the phylogenetic species concept is useful for asexually reproducing organisms, but can also be applied to sexually reproducing organisms. We would like to thank everybody for taking the time to watch, and we would also like to thank the others who made this video possible.